morning. Welcome Sham Stars. Today we are going to learn about the Lac operon. Jacob and Monod were the first to describe a transcriptionally regulated system. An operon is a unit of prokaryotic gene expression, which includes regulated structural genes and control elements recognized by the regulatory gene product. In Lac operon, here Lac refers to lactose. A polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as a operon. Now first we will talk about the components of an operon and then Lac operon. This slide shows components of an operon. Number 1 structural genes. Fragments of DNA which transcripts mRNA for polypeptide synthesis. Number 2 promoter gene. Sequence of DNA where RNA polymerase binds and initiates transcription. Operator. Third one is sequence of DNA adjacent to the promoter where specific repressor protein binds. The next component is regulator gene. Codes for the repressor protein that binds to the operator and suppresses its activity. Hence, transcription does not occur. The last component is inducer. Prevents the repressor from binding to the operator. Due to this, transcription is switched on. It may be any metabolite or hormone. Now let's talk about Lac operon. The Lac operon consists of one regulatory gene, that is I, and three structural genes Z, Y and A. Here, the term I does not refer to inducer, rather it is derived from the word inhibitor. And three structural genes Z, Y and A. Z codes for beta-galactosidase, which is primarily responsible for the hydrolysis of the disaccharide, lactose into the monomeric units, galactose and glucose. Gene Y codes for the permease, an enzyme which increases permeability of the cell to beta-galactosidase. The gene A encodes the transacetylase. Hence, all these three genes products in Lac operon are required for metabolism of lactose. In most other operons as well, the genes represent in the operon are needed together to function in the same or related metabolic pathway. This slide shows the Lac operon diagrammatically in the absence of lactose. Lactose is a substrate for enzyme beta-galactosidase and it regulates switching on and switching off of the operon, hence termed as inducer. So in the absence of lactose, repressor binds to the operator region and prevents RNA polymerase from transcribing the operon. Now this slide shows Lac operon in presence of lactose. Lactose is a substrate for enzyme beta-galactosidase. In presence of an inducer such as lactose or allolactose, the repressor is inactivated. Repressor is a protein which is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Regulation of Lac operon by repressors is referred to as the negative regulation. So children, this is all about the Lac operon, which is a unit of prokaryotic gene expression where lactose is a substrate, and this is how transcription takes place. I hope you must have understood. Tomorrow we will be talking about Human Genome Project. Thank you, Sham Stars. That's all for today. You can drop your queries on the Google Classroom. Take care.